Hi everyone, my name is Katia, and today I'll be going over the ongoing research project we have been working on, which is exploring non-motor symptoms associated with Parkinson's disease by injecting 6-hydroxydopamine into the substantia nigra of a rodent brain. To introduce this project, I'll start by talking a little bit about Parkinson's disease. So Parkinson's disease is a neurodegenerative disease that affects the nervous system due to insufficient dopamine neurons within the substantia nigra. Dopamine neurotransmitters are highly important as when they are released from the substantia nigra, they travel down the basal ganglia, and the basal ganglia helps aid in movement and body coordination. Now, due to the lack of dopamine, some motor symptoms associated with Parkinson's disease can include trembling, stiffness, and decreased speed and motion. But Parkinson's disease, or PD, doesn't only cause motor symptoms, it can also cause non-motor symptoms, which can include but are not limited to cognitive impairments, fatigue, pain, and mood disorders. Even though PD is an extremely common disease, up to 60,000 new cases per year, there is still much to understand from it. People diagnosed with PD report impaired memory, impaired learning, and poor decision making. There's a study done on humans utilizing the Iowa gambling task that found a significant difference between PD patients and the control group in decision-making impairments. So results showed PD patients doing worse on a task than the control group. But interestingly enough, there has only been one study using the rodent model. A possible cause of cognitive deficit observations is that both dopamine and the substantia nigra play a major role in reward and motivation, which both are affected in PD. Now, more than half of the people diagnosed with PD report experiencing pain. The most common pain reported is musculoskeletal, radicular, and dystonic. Past studies using rodent models that tested for allodynia, which is stimuli that does not normally cause pain, and hyperglycia, which is increased pain from stimuli that usually causes pain, through assessing thermal thresholds. And they assessed mechanical thresholds by using the mechanical paw withdrawal threshold test and found significance in hypersensitivity. Even though motor symptoms may seem more prominent than non-motor symptoms associated with Parkinson's disease, they can be just as debilitating as the motor symptoms. Yet, unfortunately, there is an underdiagnosis of non-motor symptoms associated with Parkinson's disease. In our current study, we are trying to mimic Parkinson's disease by injecting neurotoxin 6-hydroxydopamine that will cause dopaminergic cell death within the substantia nigra. This project is focused on two non-motor symptoms, which include cognitive deficits, which has an emphasis on the decision-making and also pain. We will be using the novel version of the rat gambling task to measure possible decision-making impairments. The MPWT is used to measure any possible hypersensitivity differences from their original baseline. We are using time as a factor in having half of the rodents, which are 20, that have been injected with this neurotoxin, and the control variable observed at 2 weeks, and then the rest of the rodents will be observed at 4 weeks. This is done due to most dopamine neuron deterioration at 4 weeks. We started to conduct the study by using 40 Sprague-Dawley female rats that were food restricted until they reached 85% of their weight in order to start the operant training. In operant training, rodents learned single lever press, dual lever press, until eventually they are taught the rat gambling task, or RGT for short, in the operant training. After RGT completion, they will be injected with the neurotoxin 6-hydroxydopamine or saline into the substantia nigra. We will then wait 2 weeks to test half of the rodents, and then we will wait 4 weeks to test the other half to see if there are any significant results in decision making and pain. And that is it. Thank you very much.